Hello guys, today topic is processing and quality analysis of milk. As we all know, what is the milk? About milk, as we know, it is a white liquid food produced from the mammary glands of mammals. Mammals, we have the idea that bats are mammals, domestic animals like cow, buffalo are mammals, and humans also. We women are also mammals. It is the primary source of nutrition for young ones. It consists of 87% water and 13% of solids. Most of the milk consists of 87% of water. As seen in the diagram, milk consists of 25% of calcium, 20% of phosphorus, 20% of pantothenic acid, 15% of vitamin A, 35% of riboflavin, 15% of vitamin D, 8% of potassium and 10% of niacin. It also consists of riboflavin and niacin about 35 and 10%. As going to the processing of milk, the root milk received at RMRD. RMRD means it is the raw milk section where the milk is going to be chilled by the plate heat exchanger to below 5 degrees centigrade and then stored in the raw milk storage tank till it is processed. Before it is going to be processing, the raw milk is going to be tested. In processing section, it involves in the first step involves is preheating. The milk is going to be preheating process which is done at 40 degrees centigrade. Next process involves is the clarification. Solids and other impurities are removed from the milk. Pasteurization. It is the main step involved in the processing of milk. Milk pasteurization is the process of heating milk to a predetermined temperature, means it is a fixed temperature for a specified period without recontamination during the entire process. So during the pasteurization, the milk is going to be heating at the predetermined temperature, so that the microorganisms present in the milk are going to be destroyed and the milk will be without recontamination during the entire process. It is useful to destroy harmful microorganisms. Pasteurization process are of three types. They are batch pasteurization, flash pasteurization and ultra high temperature pasteurization. Batch pasteurization, it is also known as the low temperature long time pasteurization. It involves in the low temperature, means at 63 degrees centigrade the milk is going to be heating for 30 minutes. The extended holding time causes the alteration in the milk protein structure and taste. Next involves the flash pasteurization. It is also known as the high temperature short time pasteurization, means at a high temperature for a short time the milk is going to be heated. Heat the milk to between 72 degrees centigrade to 74 degrees centigrade for a short time, means for 15 to 20 seconds only. As going to the ultra high temperature pasteurization, heat the milk to between 135 degrees centigrade to 140 degrees centigrade for 2 to 4 seconds. As we all know the Tetra packets, if we see the Tetra packets, it is going to be ultra high temperature packaging, so that the milk will remain constant without with the organisms. So the ultra high temperature is useful to the Tetra packets for a long time storage, because it is going to be heated at the 135 degrees centigrade to 140 degrees centigrade for 2 to 4 seconds. These are the three types of pasteurization methods. batch pasteurization, flash pasteurization, and ultra high temperature pasteurization. As going to the next step involves is the standardization. It is the important to standardize milk fat to ensure that you end up with a product of consistent quality in the market. Different consumers prefer different products. As we know, there are some type of packets which consists of high fat, some of low fat, some of medium fat, like red, green, and gold packet, which is of a full fat milk. As according to the partition of the milk fat, the going step involved in the processing is the standardization process. Next involves is the homogenization. Homogenization is a physical process of breakdown of the milk fat globules into tiny droplets to discourage cream separation. Tiny droplets of fat do not rise in a milk column, since reducing their sizes also increases their density in the milk. A milk homogenizer working at between 100 to 170 bars pressure. It is the homogenization step where the fat globules of the milk going to be breakdown for equally distribution in the milk. Next process involves is the chilling. After regenerative cooling of pasteurized milk, the milk is going to be chilled at the 4 degrees centigrade. After regenerative cooling of pasteurized milk, it moves to the cooling section of the PHE, where chilled water or PHE coolant lowers the temperature of pasteurized milk to 4 degrees centigrade. As we going to the tests or quality analysis of milk, it involves these types. It consists of 2 types of labs. There are fluid milk testing lab and bacteriological lab. In fluid milk testing lab, this section is well equipped for conducting all the primary tests on milk. The temperature of the room is maintained at 21 degrees centigrade. The tests conducted in this section are fat test, SNF test, acidity, alcohol test, clot on boiling test, methylene blue reduction test, heat stability and phosphatase test. SNF means solid non-fat test. Fat test. Fat test is very necessary in order to determine quality of milk, attempting of adulteration and ultimately the price of milk. Here, fat test is conducted by the Gerber's method. Next involves the solid non-fat test. Here, the solid non-fat is the constituent of milk other than water and fat. This is determined directly by using lactometer. By using the lactometer, the solid non-fat is used to be measured. The temperature of the milk should be 21 degrees centigrade. Next is acidity test. There are 2 types of acidity in milk, namely natural acidity and developed acidity. Natural acidity is due to constituents like casein, albumin, citrates, phosphates, and carbon dioxide. Developed acidity is due to the action of acid-producing microorganisms. Acidity can be measured by titrating the milk by NaOH using phenolphthalein. Next is the heat stability test. Optimum heat stability is dependent on certain ratio or balance of calcium plus magnesium ions to phosphate plus citrate ions. It is the ratio. By using this ratio, the heat stability of the milk is going to be measured. Any disturbance in the balance of either group accelerate heat coagulation. Alcohol test. Alcohol test is conducted to determine the stability of milk for condensing and sterilization processes. It indicates mineral unbalance in milk. It also aids determination of abnormal milk such as the colostrum, mashed cheese milk, etc. as we know, the colostrum means when the next involves is the methylene blue reduction test. The test is based on the dye reduction. Next is phosphatase test. Raw milk contains phosphatase enzyme. It is destroyed at the temperature necessary for the efficiency pasteurization. But when milk containing phosphatase is incubated with a p-nitrophenyl, bacteriological lab. It mainly involves in the coliform test, yeast and molds test. In bacteriological lab, coliform tests, yeast and molds swab test are conducted. Coliform test. The test for coliforms involves aliquoting a sample of milk onto an agar plate. The plate is incubated for 24 hours and any colonies formed are counted using an automated colony counter. Yeast and molds test. Small amounts of milk is taken first and it is going to be mixed with a mixture containing methylene blue and alkaline salts. The mixture is stirred while being heated. This causes fragments of mold mycelium that may be present together into a mass. The mixture is filtered and the mold mycelium retained is measured on the milk sample placed in the agar and incubated at 30 to 32 degrees centigrade for 3 to 5 days. As we all know, the swab test, it involves the swabbing of the worker hands and the equipment used for the processing etc. are going to be tested in this lab.